So protein digestion is going to start up here in our mouth, and then it's going to move into the stomach. It'll start with mechanical digestion and move on to the stomach here. Proteins will then be broken down by pepsin and HCl, which is secreted by the chief and parietal cells. Moving on to the duodenum here, we'll have enterokinase, which is going to activate trypsin, and then trypsin is going to then activate our two other zymogens here, chymotrypsin and carboxypeptidase. The S in our eye cells here are going to activate our duct and acinar cells to release bicarb from the pancreas into the duodenum here to activate all these zymogens. So once we're past the small intestine here, we'll move on. We have dipeptidase as well, which is going to break apart our dipeptides, and then aminopeptidase, which is going to break apart our di and tripeptides, but from the amine group, and we'll talk about that more later. And we have intracellular peptidase, which we'll also talk about in a little bit. Here's a breakdown of what amino acid looks like. We have our amine group on the left, our R signature here in the middle, which is what signifies what the amino acid is and what differentiates it from others. And we have our carboxyl group on the right here, which is a weak acid. So now once we're done in the intestine here, we're moving on to the enterocyte, in which we have our various transporters here, which is carry our amino acids, riding sodium into the enterocyte. Di and tripeptides are going to hitch a ride onto hydrogen through the peptide-1 transporter, again into the enterocyte, and then we have our NH3 transporter here, which is just going to maintain our sodium and hydrogen gradient here. Once we're into the enterocyte, everything's got to be broken down into an amino acid before it can go into the blood vessel. So that's where our intracellular peptidase from right here is going to do its job and cleave everything into a singular amino acid before it can be moved into the blood vessel here. We have our Na sodium potassium pump here, maintaining our sodium potassium gradient, and then we have our facilitated diffusion, carrying all our amino acids, funneling them into the blood vessel here. This is our primary active transport because it uses ATP. These are all secondary active transport because they use the concentration gradients from hydrogen and sodium. Here's a list of all the amino acids. We have 20 of them. 10 of them here on the left are the essential ones. Essential just means that your body is not going to be able to synthesize and make them on their own, so you're going to have to get them as athletes. You're going to have to get them from your food source, which as athletes, hopefully we're eating a lot of animal products. If you're getting your sources from vegan sources, you can get a lot of protein from vegan sources, but they're not going to be complete proteins with all the essential amino acids, meaning you're going to have incomplete proteins. Transamination, this is the process of synthesizing and recycling proteins. So low level here to walk through it is we have our amino acid here. We're going to break off that amine group, and it's going to be held by vitamin B6. Then we're going to have a keto acid from another amino acid, a donor amino acid, and the B6 is going to be holding that amine group, and it's going to put it onto our keto acid here to give us our new amino acid, which can then be used for whatever the body needs it to be used by. And our transaminase enzyme here is what powers this whole entire process. This happens all in the liver. This should happen about 85-90% of the time in healthy people, as opposed to deamination here, which is a breakdown of protein for energy. Not super efficient. This should only happen about 10 to 15% of the time in healthy people. Here, once again, we'll have our amine group here. They'll be carried up to the mitochondria by glutamate. We can also use glutamine if we have a lot of deamination that needs to occur, but that's using glutamine is bad. We'll talk about that later. So we have glutamate, go up to the mitochondria here, and then it will be used. It will be then hydrogen will be added to our ammonia here to make it into ammonium, which is much less toxic, and that'll then go into the urea cycle and be passed through our kidneys into our urine. And so this again should only be 10 to 15% of the time. A lot of people who are metabolically inflexible are stuck in this deamination cycle because they're using their protein for energy as opposed to their glucose, and that's super healthy. It's like throwing furniture into the fire because our proteins are valuable, so we don't want to use them for energy. Can lead to anxiety if we're using too much deamination. This glutamate right here, we can also use glutamine. Glutamine is what inhibits GABA, which is going to lead to increased anxiety in the brain. So that's again why we can't be eating so much protein. We have to be able to be metabolically flexible so that we can swing healthily between deamination and transamination. So as athletes, we got to make sure we're eating 1 gram of protein per body weight, getting all of our essential amino acids here so that we're able to go through this transamination properly and be able to feed our body so that body has enough amino acids and proteins to be able to fuel it after practice, after workouts, after games, so that we're able to do, the body's able to do whatever it needs to do to repair us and keep us ready to go.